Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rozum, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Abel Hastings from Fujifilm Diosynth Biotechnologies.
Great, thank you very much. I appreciate that introduction. As the title says, the topic of today's discussion is around the use of systematic tools for both process characterization and process validation, and expeditiously moving through those campaigns. Before I get started, though, I want to just mention that the tools that I'm going to present today and the methodology that we use is what we've learned out of our experience for process validation campaigns. So today's talk is not intended to be a sales pitch about Fujifilm, but rather to express some of the learnings that we've had over numerous process validation campaigns. And each circumstance probably is a little bit different, but the general takeaway from this, I think, is quite comprehensive with respect to lots of different programs. So in our experience, as projects move from clinical manufacturing towards process validation and ultimately towards pre-approval inspection, there's quite a transition in the methodology or the momentum behind the program itself. And this often puts mud in the water, so to speak, around the various activities. For example, the timelines associated with a project can become hyper-visible at all levels of the organization. In addition to that, any missteps that happen can become somewhat costly. And then third, and probably most importantly, the actual commercial viability of the program itself might be at stake. So it's important to maintain that focus on progression for the project. And for us, that progression really comes down to using a batch of systematic tools to strike a balance between the science of the program and the speed for launch. That balance really comes down to using science as the focus of activities and using risk-based tools to focus activities on the value-added activities that are of highest value for the project itself. In addition to that, some of the tools that we present can be useful for predicting PPQ and process validation performance, such that when we enter the actual PPQ or process validation activities themselves, we have a good assurity that we have a high likelihood of success. And then fourth, and probably most important, just having a batch of tools, a list of tools that are used by the teams, can add an element of calm momentum to the storm of all of the different activities that can happen. For Fujifilm, we've spent effort to build a suite of tools for both process characterization and process validation that we term RAPTA, that are really aimed at driving a systematic balance of both discipline and flexibility to address a wide variety of strategic directions, and as well as a wide variety of indications for various molecules. Those tools are used throughout our organization, but I'll just say it's not just about the tools themselves. Fujifilm believes quite strongly that PPQ and process characterization is not the job of the technical team or the operations team solely. It's really the job of the whole organization. So as such, we train a large portion of our staff on our methodology, from the technical team to the sales team here, in how these programs work, such that we're all speaking the same language. So as we get going on this, just the question might arise as to why you'd listen to us. So for Fujifilm, PPQ and process validation and process characterization are really more than just theory-based activities for us. It's really actually our legacy as an organization. We've built this large batch of tools for us to really maximize reliability and speed of programs, and to really drive a logic and science-based progression from GMP through PPQ. This progression or this system has been used for a wide variety of molecules, ranging from biosimilars to molecules that have breakthrough status or an accelerated status. And the tools themselves have been built with our experts that have more than 300 molecules of experience behind them. The tools have been used for a large number of campaigns, and in addition, we have quite a history of both process characterization and process validation, as well as several approved products that we work with. So the systems themselves really have been time tested. I'm a very visual person, so I always think in flowcharts, and the basic progression from GMP through to PAI can be seen in this flowchart. So this flowchart represents the progression from GMP on the left through to PAI, and begins with an understanding of both our equipment capabilities and process knowledge. I'll talk a little bit about each of the tools that we use and talk about the timeline from left to right. The first tools that we work on are actually done in parallel. It's both the process attribute map, which is intended to be a systematic process understanding, a way to drive consensus around control, as well as to identify what we call the NUDs, the new, unique, and difficult elements for each molecule that make it special. Running along parallel with that, we execute an FMEA, a risk-based tool, to help us give some preliminary classification for parameters and also to drive some data-driven decisions about equipment. The ultimate role for both of these activities is to develop a process characterization scope that is really forward-looking and aimed at manufacturability. Once process characterization is done, we advocate for a systematic and quantitative assessment of acceptable operating ranges, or AORs, against normal operating ranges, or NORs. That assessment really does predict future performance in PPQ. And then finally, the topic I won't touch on is the actual execution themselves, but we do advocate for a very systematic approach. So the first tool I'll talk about is the process attribute map. This map is a heat map of the process that shows in a consensus-driven manner the scientific logic behind the process itself. Along the horizontal axis, we see all the unit operations themselves, and on the vertical axis, we see the critical quality attributes, or release panel. And the user of this map would link these unit operations and the critical quality attributes in a heat map manner that would show either strong links or no links between the unit operation and the critical quality attribute itself. This helps to focus people's attention on the important parts of each step. Likewise, we advocate for doing the same thing for each unit operation to build parameter attribute pairs that are scientific and scientifically logical. The next tool that we'll talk about is the risk assessment itself. This falls under the umbrella RAPTA as well. And it's the risk assessment we use is a pre-populated risk assessment that's based on our experience with more than 300 molecules, but really the important part of that is what you learn from an FMEA. What you learn from an FMEA should drive what you do for process characterization. And there are three analyses that are highly valuable. The first is to look at the risk priority numbers for each unit operation in order to determine the level of characterization that's necessary based on the risk tolerance for the program itself. The second activity is to look at the sorted occurrence and detection scores for each unit operation, and that helps to determine whether there are any equipment mismatches or improvements that can be done to reduce the risk for any unit operation and improve reliability. And then finally, by sorting the severity scores by themselves, this can be highly predictive of key process parameters and critical process parameters, or KPPs and CPPs, in order to drive the scope of process characterization itself. The next activity that we talk about doing is the process characterization itself. And as we drive into process characterization, the focus of the process characterization should be about enabling manufacturing. So we believe that the process characterization design should be a forward-looking PC design that is aimed at meeting manufacturing's needs for both a reliable PPQ campaign as well as a reliable ongoing manufacturing. And there are three assumptions that one must make in order to go into a data-driven process characterization campaign. The first assumption is that the inputs for process characterization scoping really should be data-driven inputs. And this comes down to the normal operating ranges should be based on the process-specific equipment capabilities. Some capabilities are process-specific and some are process-agnostic, but those normal operating ranges based on the equipment themselves need to be considered. The second element of that is that the FMEA scores can be predictive of both key process parameters and critical process parameters. They're not always exactly aligning, but they are close. The next assumption is that there needs to be a difference between the acceptable operating range and the normal operating range in order to enable reliability. And it is our belief that that safety margin between the normal operating range and the acceptable operating range needs to be wide for key process parameters and wider still for critical process parameters. We have recommended numbers for these, but the basic premise is that we need to have a margin for error that is increasing based on the criticality. And then the third assumption is that process characterization should establish functional acceptable operating ranges for manufacturing needs. Those tools themselves are able to build a process characterization campaign that is both functional and informative. And really in wrap-up, and I'll open us up to questions after this, but in wrap-up for this, really the main point of this presentation is that systematic tools can really help expedite project teams moving from clinical through to process performance qualification. And those tools themselves need to be not just theoretical or scientific, but really be practical in nature in order to enable manufacturing to execute both the PPQ campaign and ongoing commercial manufacturing. The other important point is that emphasizing the science of the control strategy is paramount and really does help to focus the attention on teams across the organization on critical process understanding that will result in a successful PAI and successful manufacturing. And then finally, efficient and useful process characterizations are those process characterization designs are really those designs that are aimed at manufacturing reliability of the PPQ and commercial manufacturing. In other words, process characterization needs to be focused at meeting manufacturing's needs. And the statement that we like to say is that moving from GMP or from clinical manufacturing through to PPQ and on to commercial manufacturing really needs to move as fast as the science allows and no faster. So it's very important for teams to pay attention to that. So with that, I'll open it up to any questions there are from the team
great.
Or from the attendees.
Hey, Abel. So the first question is, when and how do I consider the impact of specifications on process validation campaigns?
Sure. So that's a great question. So specifications are actually the first thing that we teach when we teach process validation training here at Fujifilm. We actually believe that process performance qualification really doesn't start with the process itself, but actually starts with the specification and works backwards. So you'll see the tools that we talk about, whether it be the process attribute map or all of the other characterization itself, actually starts with the specifications in mind. Understanding where that specification comes from is quite pivotal. We like to bunch specifications into one of three categories: safety, efficacy, or manufacturing reliability. And each one of those, the sources of those specifications and the implications on manufacturing need to be considered from day one.
Okay. So to maximize process characterization, should we widen the operation range for downstream processing?
So we like to build that's a great question on the range of selection for process characterization. We believe that selecting ranges for process characterization should be based on the expected variability of the equipment. So we actually believe that we start from the expected variability in the piece of equipment and work backwards in order to build the process characterization studies themselves. So we do consider widening those ranges for any critical process parameters or key process parameters based on the scoring.
Okay. Is there any particular design of experiments approach that you recommend, and what kind of linkage experiments do you do?
So that's another great study. I myself am a very quantitative person. I enjoy math, as anyone who knows me will know. So it's very easy to jump into the statistical bandwagon and jump onto the latest DOE or most complex DOE. To combat that, Fujifilm has actually built a decision tree that helps the user or the designer for a process characterization campaign to select a full factorial design when necessary or a response surface when necessary. And I'll say that I'm always surprised at how often a one factor at a time or an OFAT study is actually the most effective characterization methodology to get at the root understanding for a parameter attribute pair. So there isn't one solution. I would really caution the user to select the right study for the right application. If there are linkages, and that was a great subset to this question, if there are linkages, that's a great opportunity to employ a DOE to cover the design space more fully to look for secondary or tertiary interactions. So if there aren't linkages, that's a great opportunity to do a one factor at a time. If there are linkages, great opportunity to select a multifactorial or more complex DOE.
So I think this is going to be the last question. If we didn't get to your any question you typed from here forward and previously will be sent to Abel directly, and he can follow up with you via email. So the last question is, I am working on a clinical program. When should I begin using these tools to find balance between overburdening the project and the risk of doing rework?
Sure. So I would highly recommend using both the FMEA tool and the process attribute map relatively early in the program. The important point for that, or the important element of that, is that risk assessments and FMEAs, as well as the process attribute map, don't need to be a fully baked cake in order for you to use them. They're intended to be both iterative processes such that we allow ourselves to both capture our current understanding and identify gaps in our understanding for the next phase of development. So I really highly recommend for teams to use tools such as the process attribute map or the FMEA from day one in a simplistic manner, and expect that understanding and the depth for those tools will continue to grow as projects progress from clinical through to PPQ.
Okay. Thank you, Abel.
Thank you very much. I appreciate the opportunity.
So thank you to our audience also for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcast. Look for those announcements in your inbox. Have a good day. Goodbye.